Hello, good evening, everyone, and thanks for being here. A very warm welcome to today's webinar, Winning in the Face of High Soybean Prices. My name is Arun, and I am the marketing manager with BIOMIN in India. I will chair and moderate the sessions for you today. This webinar comes in the backdrop of unprecedented rise in soybean rates. Just to give you a context, soybean meal rates have increased from rupees 35 to rupees 93 a kilo in a matter of 5 months. This is what the report from Business Standard says. So to help the industry, poultry and livestock industry, we are organizing this webinar. And for this webinar, we have brought in two experts, Dr. Neil Gannon and Mr. Michele Muccio, both of them my colleagues from BIOMIN. The first speaker today is Dr. Neil Gannon. Born in Sydney, Australia, he holds a PhD from University of Sydney. He joined BIOMIN in 2011 as Managing Director for Australia, and from 2015 commenced his role as Regional Product Manager, Gut Performance for entire Asia. So today Neil will handle the topic how to optimize use of soybean meal and alternatives in the current price environment. So before I hand over to Neil, I got a suggestion from Neil to run a quick poll so that all the attendees get involved in this webinar. So I will now run a quick poll. Give me a second. Yeah, you should be seeing the poll in your screen now. I will read out it for you. Do you have reliable information on the nutrient content of the ingredients you use in your rations? So the options are yes for most products, yes, but only for the products used in large amounts. Not really, it is bit out of date. No, there is little information. So I think most people have already chosen an option which I can see now. I will give you a few more seconds before I close this poll. Thank you everyone for your active participation. I think it's very interesting to see the poll results. It's quite balanced. Well, I stop the poll now. So I will share the results now. So 42% have given their opinion like yes for most products. 46% yes, but only for the products used in large amounts. 4% not really. It's a bit out of date. And the last option is no, there is little information available. So, Dr. Neil, do you find the results interesting? The stage is yours now, and we are eager to listen to you.
Yes, thank you, Arun. Very interesting. I wondered if you could just stop sharing the poll results because I'm not able to share my screen at the moment. But I'm very pleased to see that most people actually have a number of, believe they have good detailed information on their raw materials, which is great. So very few people were in the know or limited. And this is really good to hear because it's the essence of my presentation. In terms of making sure that people have good nutrient information, because without that, it's very difficult to assess how to use these alternatives to soybean meal and even to use soybean meal to its best efficiency when the prices, as you indicated, are very high. So yeah, thank you for that. And another warm welcome to all of our attendees, and thank you for giving up your valuable time today. What I would like to give the overview is just quickly review, and then again, most of the people here are formulators or nutritionists or understand why we use soybean meal in diets, but I wanted to put it in a bit more context. because then I can reaffirm the importance of knowing the nutrient specifications of our ingredients. And from the poll, I'm very pleased to see that most people do understand that. And then to help us position the use of soybean meal and alternatives, introduce this concept of metabolic loss, and then quickly identify some strategies for ensuring that we will optimize all the ingredients available in a feed mill. And as Arun indicated, please ask questions at any time in the chat box. I know we have a very short session today, this afternoon, this morning, depending on where you are, but please ask questions and we'll try and get through those in the discussion time at the end of this webinar. As you know, that, and this is just some data from the US, looking at the nutritional value of US soybean meal compared to Argentina and Brazil. And it's also, yes, it was a report by the US and they were trying to obviously sell more US soybean meal. But if we just take a look at the big numbers, soybean meal is used because it's very high in protein, 46, 47% protein. And it has a very high concentration of lysine, and lysine is one of the most important amino acids for all animal species, particularly poultry. And so you can see through here, and again, those that understand soybean meal will understand these numbers. It has a little bit of phosphorus in there as well, which is important, and calcium. But when we look at energy, again, not too worried at this stage about the differences in the energy value between the Argentinian, the Brazil, or the US, but it is a very good source of energy, and the energy variation is largely contingent on how much oil is left after the processing. So we use soybean meal for its energy and its amino acid content or protein content, and therefore it's quite a valid source. But even we think that soybean meal is quite a uniform product. It comes in a boat all the time. But when we look at some of this data here, where this is just looking at the crude protein and how crude protein content has an impact on the lysine content, and as the crude protein level goes up, so does the lysine content. And because we're trying to supply lysine to the bird, if we don't know what the crude protein content is, then it's very difficult to estimate in our formulation programs what is the actual lysine content. And again, there was a USA report, but what it is showing is that US soybean meal is probably more consistent than some of the other Argentinian and Brazil sources. So I'm trying to introduce the concept of why we need to have good information, because if you look across here, you might have this particular batch of soybean meal in your plant with a low crude protein, and this is on a dry matter basis, so these numbers look higher than the 46 or 47%, which was an as-fed. Or you might have a soybean meal with very high protein, which has a higher lysine. So again, simple measures such as protein content will help us understand the amino acid content as well, but it only goes so far. Again, from this report, but there's many of other examples in the literature, and this one was quite interesting because it now includes Indian soybean meal. And when you start looking at the ileal digestibility, and I won't go into too much terminology around this, but what we're trying to look at is how effective is the lysine in the soybean meal utilized by the bird, and in this case, the broiler. And these studies were done in New Zealand at Massey University. And you can see that just looking at the numbers here, there's big variability depending on which amino acid you're looking at, depending on where the source is. And again, if your supplier of your soybean meal has switched from Indian to Brazilian or USA and hasn't told you, you might be formulating on the wrong nutrient specification. So again, even though we think soybean meal is a very consistent product, the results are indicating that it's quite variable. I also want to introduce a concept that you probably all know, this barrel theory, the limiting nutrient approach. And this was a very old approach, first coined in 1828. But what it's trying to indicate is that if we look at, say, limiting amino acids, the animal can only perform to the lowest common denominator. And in this particular case, if you've got a diet, largely based on your soybean meal, contributing some lysine, and lysine is low, the birds won't perform, whether they're broilers or layers, won't perform to expectations until you get the lysine level up to where it's required. And this is most effectively done by adding synthetic amino acid to the diet. And most nutritionists understand that the ratios here. But what happens when we add lysine is that then we come into a second limiting amino acid. In this case, it might be threonine or cysteine or methionine. And so you keep needing to increase the limiting or remove the effect of the limiting amino acid. And if you just hold that thought for a bit longer, I'll show you why that can be important for poultry. Some of the ingredients that are used in poultry feeds across the Asia Pacific region, and maybe not all of these are available in India, and there's probably even more that are available in India. But you can see from this short list, most of the cereals are at the top here. Then we go into the various protein meals, and then we go into some of the specialist byproducts, and then obviously the minerals and the vitamins and minerals and premix and pure amino acids. So very quickly you can see that there's a lot of raw materials that are available to our formulators and nutritionists to use. And if each one of these has a variability, like we talked about with the soybean meal, then how do we actually predict with our very reliable formulation programs what the diets will be and how the animals will perform? And this is where I want to start to introduce about the various alternative raw materials. Throughout this talk, I'll just be mindful I'm showing some data, but we know that these are just average values or typical values within a country. And these are really just for illustration purposes, and do not use these in your own formulation because, again, you have to know what the source is in your country. But what I'm trying to show here is that if we look at the energy values, and there are some variations, and that's for these byproducts and things, is largely related to how much oil is left. But you can see there's a big variability in the protein content as well as the fat. But more importantly, for some of these alternative raw materials being proposed for soybean meal, the problem comes in with the fiber level. Very high fiber levels in things like palm kernel meal, copra, but even dried distillers grains. And then we talk about the lysine content, and this doesn't even start to consider the digestibility of lysine, because we know that there can be big variations in that digestibility, even though the total lysine might look relevant. And then, of course, the moisture content. And as we start to get into some products, very high in moisture can run the risk of storage issues in terms of mold development, which leads into mycotoxin risk management, which Michele would talk about after my seminar. So along with all of the nutrient variability, because of different processing, we also run the risk of mycotoxins. There can be some chemical residues, and this can have a big impact on the food chain. There might also be some quarantine risk. The governments of your countries may not allow you to bring some of these products in because of risk of either weed seeds or GMOs or a whole lot of things. But more importantly, from a poultry perspective, the microbiological contamination, E. coli, salmonella, particularly for compounds that are just dried out on the roads or the fields. So again, just because the nutrient content might look good, we have to be mindful of some of these other potential hazards that can occur. In terms of the ingredient variability, why do we have this variability? Why can't we just take one value for soybean meal and use it all around, or for corn, or for our rice? And the problem is that we're dealing with, a biological system. The crops are grown under different weather conditions, soil types. They could be different stages of maturity when they were harvested, different fertilizer and irrigation programs. And then we start to look at these co-products, these byproducts, such as even soybean meal as a byproduct, because we humans want the oil from it. Dried distillers grain in particular. Depending on the dryer designs and the speed of drying and the temperature, you can get big differences. Even with the fermentation process of the corn, you can get big differences in the concentration of nutrients. And often color is deemed a good indicator of your dried distiller grain quality. But sometimes it's not, people don't look at that, and they just take a book value or a standard value they've used all the time without relating to the raw material sampling process within their feed mill. So again, just a little bit of a reminder to look at the quality and the visual aspect of your ingredients to make sure that what you're formulating is representative of what you've actually got in your feed mill. Again, to illustrate, we've talked about the variability in soybean meal, and this is some old data, but it illustrates the point. And with corn, we have a big variation with corn. And this could be representative of India, it could be representative of imported product, it doesn't really matter. All raw materials, when you look at this, will have this variability. And if you look within a particular month, you can see that there's going to be big variability in the protein content of soybean meal, whether it's 46% or up to 50%. And you have to be able to manage this sort of number because if you've got soybean meal in your diet at 25%, this variation could lead to a difference of 1% in the total protein of your diet and all the corresponding impact of the amino acids. So again, variability of raw materials is really quite important. Some old data, again, this came from Keith Benke at KSU in the US, but it illustrates the purposes of how crude protein variability can vary quite dramatically. And then when you go in and you can change some processes at plants, again, in terms of lysine, you can have a material shift in the availability of lysine because of some changes in the diet, in the process. So again, as a nutritionist and a formulator and a purchasing person, you need to have good relationships with your supplier of your ingredients to make sure you understand any changes that might have happened in the background of the product, even though it's still called the same name. Again, for illustration purposes, this is what goes on the back of a formulation program. But when we look at the metabolizable energy, and I apologize, this is still in megajoules, not kilocalories, but it's just for illustration. And we can see that the grains typically have a low energy content, whereas oils and tallows have a very high content, and molasses has a lower value than grains. What happens though, when you look at the dollars per megajoule, how much is each of these contributing? You can see that there's a big variation depending on the energy content and the price that you're paying for it. And this is the same analysis that goes on in the back of your formulation program when soybean meal prices go high. And what we can see is that even though tallow is very high in energy, it can be used to boost the energy content if you're going to use some other raw materials with a low energy content. So tallow, even though it's more expensive per megajoule, you don't use much of it, and it can save some cost because you don't have to use as much of your grains. And again, this is what formulators do on a regular basis. The same thing can be done as the formulation package does when we look at lysine. And so when you look at the dollars per kilogram of available lysine, it's very expensive to add lysine in the form of your cereals, but because the cereals make up more than 50% of your diet, they do contribute a significant amount of lysine at the same time they're supplying energy. But as we can see, soybean meal is a very cost-effective way of adding available lysine. And more effectively cheaper is synthetic amino acids. And again, this is where one strategy in high soybean meal prices is to use more strategically synthetic amino acids to try and balance the diet, and also maybe supplement. If you're going to reduce your soybean meal content and use some byproducts, you would make better use of those byproducts by supplementing with synthetic amino acids. If you remember back to my barrel theory, the problem with this approach of increasing your alternative raw materials to get to a minimum lysine is that you can look at, for example, phenylalanine and tyrosine here. The levels are now very high, and these are the essential amino acids, but we have to remember that in protein, there are also the non-essential amino acids, the glycines and other ammonia, nitrogen-containing compounds. And if you keep increasing your protein, alternative proteins, to get to the lysine level, you may inadvertently be lifting the total amino acid content of the diet. And in doing this, there may be too many other amino acids in surplus to the animal's requirement, and these amino acids may be undigested and end up in the hindgut and run the risk of inducing necrotic enteritis or causing ammonia issues in your sheds because there's too much nitrogen being excreted. So again, this helps with the support of synthetic amino acids to try and balance the total nitrogen content in the diet, not just the essential amino acids. When we move into quality, and quality means different things to different people, but from what we're trying to do as nutritionists is we're trying to formulate on kilocalories, we're looking at available amino acids, we're looking at fat and fiber, we're looking at phytate levels so we can add our enzymes, and we're also considering, in the case of pelleting diets, some of the palatability indices, and maybe also looking at the palatability factors. Some of these byproducts might be very bitter, such as rapeseed meal, could have some impact on unliked odors in the eggs. So we have to be balancing all of these things. But when we get the farm specifications, What we typically get for grains and some of the other byproducts is that this is just dried distiller's grain. This is your broken rice. It gives you the moisture, but often it might even not even have some of these trash and foreign material things. But none of this material, which comes on the quality report, is actually relevant to the nutritionist. And this goes back to my original poll question. We have to make sure we know all of the information about our raw materials, because the farm quality parameters have no bearing on the feeding quality. So what we're buying and trading our raw materials on is of little value in most cases to our nutritionists. And there's many services out there, near infrared services, analytical services, book values, which will, if you're getting a new raw material that you're not familiar with, this will give you a start, but there's not too many ways of measuring the metabolizable energy of a raw material, because this involves extensive and expensive testing with live animals. I want to now move into the concept of nutrient sparing. And again, just to keep it quick and simple, if we look at a nutrient utilization in the perfect world, feed goes in a large amount, we get some digestion, and you end up with a very small amount of feces. Along the way, the bird needs a lot of energy and nutrients for maintenance, and then hopefully maintenance is very small, with most of the nutrients going into growth and reproduction, whether that's the meat, the milk, or the egg. And hopefully there's a very small amount of unproductive purposes or metabolic losses, which is just the inherent inefficiency in our systems. So this is what we'd like to see in the perfect world. But unfortunately, in the real world, maintenance is still the same. The unproductive purposes or the metabolic losses, because of impacts of mycotoxins, anti-nutritional factors, phytates, etc., this becomes larger, which means it takes away from what you have available for growth and reproduction. And this is the nutrient sparing concept. What we want to try and do is minimize this particular contribution. So the working definition of nutrient sparing is that simply sparing means freeing up or saving from a particular purpose. Therefore, nutrient sparing is applying strategies to optimize the gastrointestinal tract and metabolic environment to ensure that all of the nutrients we're feeding to our animals are digested, absorbed, and utilized for productive purposes. And when we start looking about some of these alternative raw materials, they may well be digested and absorbed, but the raw materials, the nutrients, are not utilized. They're often just straight excreted. And therefore, we have less nutrients being diverted into these unproductive processes. What are the key causes of metabolic losses? Well, typically it could be formulation issues. We just don't have the right information on our matrix for the raw materials we're using, and therefore we under or overestimate. It could be the type and quality of the raw materials used. And of course, there's also the stress and immune system activation. And we know that stress is a large part of the stresses in the gut are a large consumer of energy and amino acids. But this could be a bacterial stress, could be heat stress, but also, as Michele will talk about, the impact of mycotoxins. And of course, there's going to be combinations and synergisms of all of these. So when we start looking at the importance of the gastrointestinal tract and nutrient utilization, I like to use this sort of illustration. And if you imagine that this amount of feed down here is what the bird gets every day, and there's a limited daily nutrient supply. And then you've got all these processes, a bit like all these dogs lining up to eat something. And the first dog in the row is the gut, because the gut gets first look at the nutrients, and so it's going to use what it needs, particularly for immune responses. And then we can get on to maintenance. Energy and amino acid can go to maintenance, and then we've got heat regulation, activity, and all these other processes. And somewhere down the back, these dogs, whatever nutrients are spared, available for growth and reproduction, can be used. And so when we're feeding our animals and looking at our raw materials and our byproducts, we want to make sure that all these dogs are using as less feed as possible, so that we have as much food going to the growth and reproduction of dogs. So how can we do that? Well, one example is to use phytogenics, and we know that phytogenics are simply phytogenic feed additives. They can be herbs or spices, essential oils, or extracts. And one example that we have in the BIOMIN stable is Digesterom DC, and this is a duplex capsule formulation where we have a natural menthol-based outer capsule and then inside the capsule, protected from the harsh conditions of pelleting and the digestive system, the proventriculus, we have the gut microbiota modulation modules, and this is carvacrol, thymol, and carvone based. There are some other ingredients, and then also we have some gut protection components in the product as well, along the lines of cinnamon bark, oak bark, and grapeseed. So how can we use digesterom to help with this this down-specking and feed cost formulation under high feed prices? Well, typically, if we just assume that we have normal feed cost, we have normal performance, this is what you would have. Whenever you add an additive, such as digesterom, your cost goes up. If you look at this red line, if you can see that on your screens, and then, but by adding that digesterom, you would expect higher performance. So you have an improvement here compared to the normal performance. But the problem is that you've now already got a high feed cost and you're now adding additional cost. So what can we do about it? Well, again, we don't have time to go through in too much detail, but we know that digesterom has an improvement in terms of utilization of nutrients, particularly byproducts. We know that it optimizes the gastrointestinal environment, and so therefore it's nutrient sparing. So what we can do is we can down specify the diet to actually save some money and still add the digesterom in, and we can obtain normal performance, or we can keep the cost the same. So optimize the diets, allowing for various down-specking, keep the diets the same, but get higher performance. And so this is something that we'd need to talk one-on-one with you from the BIOMIN perspective. This is really suitable for broilers, probably not so much in layers where you really want to optimize feed intake, but it's a strategy that can be used, and please reach out to your BIOMIN representative. These slides will be available and the video you can go back to look at them, and I won't go through too much, but what we wanted to show here is that we've had a standard diet, T1, 2, and 3, and we had some antibiotics, growth promoters, or we had the digesterom or the control without. And then we reformulated each of those diets down to 95% of the specification and added digesterom in this particular case. And they're corn soybean meal diets. And we fed them to broilers over the standard phases. And what I wanted to show here is that when you look at the normal diets, the phytogenic feed additive improved the growth rate compared to the antibiotic growth promoters or the normal diet. And when you down-spec the diet to 95% of this, not surprisingly, you expected down-spec in the growth rate. But what was interesting in this study was that if our estimate of this down-spec and the contribution of digesterom is correct, then we should be able to return the phytogenic level to the control. And this is exactly what happened. So this was the control level. We down-spec'd it, and we could almost return the performance to the control diet. So showing that digesterom over the top is effective, but also it can help with this nutrient sparing approach. And we can also see in terms of the FCR and the normal diets, the digesterom treatment group had the best FCR. Not surprisingly, if you down-spec the nutrients, the FCR gets worse. But again, the phytogenic feed supplementation improved the FCR. Again, we look at the production index across the different treatments, and the production index is an indication of body weight by survival rate and feed conversion. And you can see that the phytogenic feed supplementation improved productivity. So the take-home message from these are that supplementation of standard diets can improve growth rate and FCR when you put digesterom in. Down specification of a diet by itself reduces broiler performance, but because of the nutrient sparing effect, you can do some supplementation with the diet. So in conclusion, soybean meal is a valuable protein source. It's widely used around the world. Alternative raw materials are possible, but the details, you need to have the details of the raw material composition. You have to be careful consideration of the various factors, mycotoxins, chemical or other microbiological contaminations. And reducing the impact of metabolic losses is always important, particularly when feed prices are high and meat and egg prices are low. So the use of phytogenics such as digesterom may assist in ensuring dietary ingredients are fully utilised by the bird. And as I said, time limits a full description of these concepts, but please reach out to your local BIOMIN representative for further information. Thank you, Arun. Back to you.
Thanks a lot, Dr. Neil. That's Dr. Neil for you. He's coming with over 30 years experience. It is always very informative listening to you. I guess you covered wide range of highly specialized points, and I'm sure that this will be really helpful to the formulators and nutritionists. And thank you, Neil. And to the audience, I would also like to remind that there is a question section in this GoToWebinar. So if you have any questions to Dr. Neil or Michele, you can please key in your questions. So we will try to cover as much questions as possible towards the end of this webinar. And one more general announcement is, of course, if you just in case if you miss any key points, you don't worry about that, because we got you covered, and we'll be sending you the recorded video as well as the presentation slides to you in just 1 or 2 days. With that, I would now like to introduce our next speaker. Our next speaker is Michele Muccio. Born in Rome, Italy, he holds a master's degree in biotechnology. He joined BIOMIN in 2015 as product manager for Mycotoxin. And in 2019, he took over the role of regional product manager for mycotoxin risk in Asia. Before I hand over to Michele, I would like to run the second and last poll of this webinar to get your insights on mycotoxin risk management. Before I hand to you, Michele, I will run a poll. So it should be visible on your screen now. How do you manage mycotoxins? So how do you manage? The options are: only control aflatoxins with a simple binder. Periodically, mycotoxins are a seasonal problem. I don't. Poultry are resistant to them. And the last option is with a broad spectrum deactivator. So answers are coming in fast. I would wait for a few seconds, Michele, before I hand over to you. I'm closing the poll now. So here are the results. 54% have chosen they manage mycotoxins with a broad spectrum deactivator. And 25% have said they manage mycotoxin periodically. Mycotoxins are a seasonal problem. And 21% have chosen only control aflatoxins with a simple binder. And 0%, I don't. Poultry are resistant to them. So none have chosen that option. So Michele, this is your area of expertise. I will now hand over the stage to you. What are your comments? And please also move on to your presentation. I'm sharing the view now.
Yes, great to hear that 54% of our audience is actually using a broad spectrum deactivator, and I'm actually happy to hear that like no one is thinking that poultry is completely resistant to mycotoxin. And for the people that are still focusing on using a simple binder, and for the audience that is only controlling mycotoxin periodically, I'm hoping that I will manage to change your mind with this presentation. So welcome everyone to all attendees from my side as well, and thanks Aaron for the introduction, and also thanks to everyone in this webinar for dedicating some of your time to us. As Aaron already said, please ask questions in the chat box below, and we will follow up with you. So in my presentation, as you can see from the title, I will pick up a little bit from Neil's talk and discuss talk about these alternative raw materials from a mycotoxin risk management standpoint. I have divided my talk into 3 sections. So at the beginning, I will address what are the problem and the solutions of these alternative raw materials and why do we need to take care of mycotoxins. Afterwards, in the second part of my talk, I will show a little bit of the results of a survey that was done in India and where many customers have sent some of the alternative raw materials which are commonly used, and I will go through them and see the contamination and show you the contamination. And in the last part of my talk, I would discuss why actually using Mycofix makes sense. Okay, I'm not going to focus too much on the product formulation because you know that already, but I want to show you why it makes sense to use this product. Without further ado, I'm starting with the first part. So what are the problems when it comes to alternative raw materials? I think Neil already made a brilliant summary, but I just took 3 points for myself. So of course we are dealing with the price of soybean meal and also other commodities like corn that are going up. And of course the industry needs to look for cost-effective solutions to compensate this. Also, there is a need to maintain a good nutritional value in the diets and ensure good productivity. So actually point number 1 and number 2, they're strictly related. And the other problem that comes with alternative raw materials is the presence of anti-nutrients and other components like mycotoxins, which are affecting the health and the productivity. So what are the solutions to this problem? Again, I pointed out 3 quick fixes. So the first one is, of course, to perform an accurate selection of those alternative raw materials. So we need to make sure that we are selecting good materials from a quality standpoint and also a nutritional value standpoint. So we need to make sure that those materials make sense in the diet. Also, we need to test for mycotoxins, so we need to test for anti-nutrients as many as we can, and we need to test for mycotoxins because some of these commodities are highly contaminated by mycotoxins. And testing is something that has to be done consistently, is something very important, and ideally many people, they tend to test maybe at the end of the season or something, but test has to be consistent. Ideally, every single batch that enters the farm or enters the feed mill should be tested for mycotoxins, because the goal is to respond to the problem before it reaches the animal. So working on prevention rather than responding to the problem. So testing needs to become really the bread and butter of practices, especially when we're using those materials which could be highly contaminated. And the third point, how to solve this issue, is to use scientifically validated feed additives for good nutrient conversion. And you've seen before the digesterone, and full mycotoxin control. So also full broad spectrum products that can deactivate those mycotoxins. So why do we need to control mycotoxins? Well, there is plenty of evidence on the effects on animal performance at field doses. So we are not talking usually about the high levels that we see on peer-reviewed papers, but there is more and more evidence of these negative effects happening at field doses, at doses that you find every day in the feed. And also we need to keep in mind that the mycotoxin prevalence is increasing worldwide. We also need to keep in mind that mycotoxin disrupt the gut health and the immunity. Gut health and immunity go together because we have a direct effect on the feed intake and the conversion. And also we have, the gut is also responsible for 70% of the immunity. So we also have decreased vaccine and decreased efficiency of treatments. We also need to keep in mind that mycotoxins are predisposing factors for several pathologies, and they make pathogens more aggressive. And last but not least, mycotoxins come in cocktails. This is always true. Feeds are contaminated always by multiple mycotoxins, and they undertake synergistic interactions where the toxicity of one mycotoxin is increased by the presence of others. I mentioned before that the mycotoxin occurrence is increasing worldwide. You probably know about this old FAO report that estimated that 25% of world crops are contaminated by mycotoxins. Well, new publications are actually proving that this is wrong. And not only that this is wrong, but it also proves that this data, no one had an idea where it came from, and the real picture is close to 60 and 80% to 80%. So 60 to 80% of worldwide crops are actually contaminated with mycotoxins, and we have actually achieved this number also thanks to the great advances that have been made in mycotoxin analysis. What is also stressed in recent research is the fact that animals and humans are exposed to cocktails of mycotoxins. So finally, there is an attention of the scientific community and of the regulatory of the government authorities and everything to the effect of cocktails of mycotoxins on animal and human health. I mentioned before the importance of synergism, and I mentioned before how it's very rare to find commodities contaminated only by one mycotoxins. Well, synergism is, of course, the condition when the toxicity of one mycotoxin is increased by the presence of other, and it's actually the rule and not the exception, because it goes hand in hand with the concept of co-contamination. We need to keep in mind, if we have a look on this graph on the left-hand side, that there are already numerous synergistic interactions that have been established in the literature, and it is proven how some of those mycotoxins, they work together to decrease performance and make pathologies more severe. Synergism is also very important because we need to keep in mind that feed are contaminated by more than one fungi simultaneously, and more fungi produce different mycotoxins. So again, we come back to the fact that the co-contamination is the rule and not the exception. The same fungus also can produce different mycotoxins simultaneously. Mycotoxins are chemically stable and they resist to processing methods. So you will find mycotoxins in your finished feed. And also that not only we have mycotoxins in the feed, but we also have other contaminants. We have heavy metals, we have pesticides. I know pesticides are a big topic in India. We also have bacteria, and we have other toxins that can play a role. So all these factors together, they can increase the toxicity of feed. When it comes to the interaction of mycotoxins with pathogens that I mentioned before, I want to bring to your attention this graph is from a publication from 1984, where we have on the bottom here the increasing concentration of mycotoxins, and on the axis here we can see all the on the x-axis we see all the conditions that are occurring when the mycotoxin contamination increases. And you can see that when increases with increasing levels of mycotoxins, we have episodes like immune suppression, metabolic disorders, but they become a lot more severe if we add a pathogen to the mixture. And some of the consequences of the immune suppression are, of course, the increased risk of infections, the more severe disease processes, therapies that become more difficult, and also impair vaccination response. And this has, of course, a direct impact on productivity, and also a direct factor for an increase in cost. This slide here summarizes everything that I mentioned before, so the interaction of gut health, immune system. So if you have a look on, and this is a slide that I took from a publication from Antonisen, and you've probably seen these slides many other times in webinars. If you look on the left-hand side, you can see this is the healthy gut, okay, with our villi nice and structured, the tight junctions are in there, so the permeability is ensured. But when a mycotoxin arrives, in this case, deoxynivalenol, we see the effect on the villi, so the villi atrophy, the villi fusion, and this, of course, means less nutrients. The gut is absorbing less nutrients, so we are wasting feed. And also we have the disruption of tight junctions, which is something caused by multiple mycotoxins, including DON. When we are disrupting the tight junctions, of course, we are losing this gut wall capability, and we are opening the door to all kinds of stressors, so pathogens, other mycotoxins, and so on. And not only things can go in, also things can go out. So we can assist to phenomena like the leakage of plasma amino acids, that they will start going to the gut, and then they will become the substrate for the growth of bacteria. And in many cases, it's the pathogen bacteria that grow. And when the pathogen bacteria using those nutrients are outgrowing the beneficial bacteria, we assist to dysbiosis, and then we assist to necrotic enteritis, coccidiosis, and all these problems that we know well in the industry. So hopefully I gave you a little bit of a brief overview of why we need to care about mycotoxins. And I want to go into the second part of my presentation by showing some of those of the results from Indian samples. So this is a survey that we have done in the country. We have analyzed some of the commonly used alternative raw materials, which you can find summarized in this table on the left-hand side. This analysis was a mix of ELISA and CMSMS, so state-of-the-art mycotoxin analysis. And I can conclude really briefly, just to give you a summary, that the sample contained, on average, a medium to high contamination profile, and also that all samples had a high potential for synergism. Because if you look here on the right-hand side of the table, you will basically see that no samples, with maybe the exception of this jowar or sorghum, was contaminated by only one mycotoxin. And even here, I only included the mycotoxins which were present at high levels. So even in the jowar, the Zen was high, but there were also other mycotoxins. And you can see that in most of these other samples, all these mycotoxins, they're present together at levels which could influence performance. Importantly to mention is that nearly all the feed samples that we have received, actually pretty much all of them except for like a few, they were for layer and breeders birds. So we're talking about long-lived birds with a long production cycle. And we are talking about birds that represent an investment, and we need to protect this investment. Digging into the mycotoxin survey, I split those commodities into similar clusters, and I'm using here a color scale to indicate the risk. So yellow stands for low risk, medium stands for medium risk, of course, and orange stands for medium risk, and red stands for high risk. And of course, the low, medium, and high is decided based on our internal guidelines. If we have a look at this rice byproduct, again, I'm reporting the average. I'm reporting the average of the samples they received. But bear in mind that the average has many limitations. So the average does not look into single farms. And when it comes to mycotoxin management, it's important to stress out that we need to look into single farms. And also the average, the major limitation of the average is that it basically takes all the samples together, and samples are basically coming from the farm, and we don't know whether these samples have been taken in a representative way. So they can be biased from the way the sample is taken. So having a look at broken rice, rice polish, and rice bran, we can see a general contamination profile, which I consider medium, with the several mycotoxins present at medium levels. Of course, I didn't mention before, these are the abbreviation that we use for mycotoxins. So aflatoxin, zen-zeralenol, deoxynivalenol, T2 toxin, fumonisins, and aflatoxin A. My advice for who uses these commodities is to monitor the spikes in aflatoxin, deoxynivalenol, and fumonisins. Because even though the average is a medium, we receive many samples with high levels. So again, individual farms should always keep an eye on that. And actually, this advice is shared among all the samples that I'm going to show. Maize byproducts that I used, that have been sent, so DDGS and corn gluten meal. Again, DDGS, the samples that I received from India, they all contained a medium, let's say the average is a medium contamination profile. And the CGM, corn gluten meal, is also medium with the high spikes in aflatoxin. Overall, if we need to take it as a summary, I would say that this profile is medium high. And also my advice is not to trust DDGS and keep monitoring DDGS for all mycotoxins. Because again, I'm showing you here that the average of these few samples that we received is medium, but we know from an experience point of view, and we know from results from other regions, that DDGS is usually very high in mycotoxins. And even some of the samples that I received here, they had very, very high levels of mycotoxins, especially fumonisins and deoxynivalenol. So when you're using corn, maize byproducts, always keep an eye for the storage mycotoxins and keep an eye on the level of fumonisins, which can be present at really high levels in those samples. And fumonisins are a known contaminants of maize and maize byproducts. I have a few cakes in here, so groundnut cake, mustard cake, rapeseed cake, and sunflower cake. And again, as I mentioned before, the general contamination profile is medium to high. And some of the samples that I received, they were really heavily contaminated. So most of the mycotoxins that I've seen of concern are the storage mycotoxins, like deoxynivalenol and fumonisin, which were present at really high level in some of the samples. But also, let's not forget about aflatoxin, zearalenone, and also trichotesins in general, which can still represent an issue. They can work synergistically with each other. And again, they came up in high levels in some of those some of those samples. Other alternative raw materials that I received in the surveys were jowar or sorghum and also pearl millet. And again, as for before, I keep the general contamination profile as medium high, as some of these materials were heavily contaminated, and all the jowar samples that we received contain actually a high amount of zearalenone. So even the average here looks high. Aflatoxin, zearalenone, and trichotesins might also represent an issue in these commodities. So if you are a layer and a breeder producer, or if you manufacture feed for layers and breeders, be cautious, especially when using high amount of sorghum in the diet. And my advice is always to monitor and apply a proper counteraction strategy to mitigate the effects of those mycotoxins. Last slide to wrap up this survey part is to remind you that the mycotoxin contamination is a dynamic process. So there is no season. There is no mycotoxin season or something. Mycotoxins contamination is fluctuating throughout the years, and it varies from batch to batch, which is why, again, we need to test. This slide that I'm showing you before, it's the contamination of deoxynivalenol that has been followed in one customer throughout a full year. And as you can see, yes, you have weeks with low contaminations, but they are usually followed by weeks with high spikes. So imagine you were that customer, and imagine you took a sample over here around mid-July, and you will think, oh, okay, it's actually low risk. I only have 200 ppb of deoxynivalenol, so I don't need a mycotoxin counteraction strategy, or I can use a chip binder. But 2 weeks later, it goes from up to 1,000 and even 1,500. So mycotoxin contamination is dynamic. It's always related to the batches that are entering the farm or the feed mill. So that's why it's important to invest into a mycotoxin counteraction as an insurance policy and to use this throughout the whole year and not to only focus on some spikes. Try to anticipate the problem rather than responding to it. This would be the secret for both productivity and for a healthier use of those alternative raw materials. This slide actually would be an amazing hookup for the next part of the presentation that I want to stress on. So is why using Mycofix makes sense. Okay, and we always hear Mycofix is expensive, no need for mycotoxin counteraction for the whole period. But in the next 2 slides, I just plugged in some numbers just to make sure, just to show you how actually it makes sense to have a good product that you know it can do the job. And let me show you an example with layers. Okay, so I ran some calculation through a calculator that was developed by a colleague of mine. And although it says euro over here, I couldn't get rid of the euro because it's embedded into the calculation, but the prices are actually in Indian rupee. So assuming a Mycofix dosage of half a kilo, and yes, you can use as little as half a kilo. Mycofix has a broad range, a very flexible dosage from half a kilo to 2.5. And I chose half a kilo, the minimum contamination, because although I mentioned that some of those materials, they contain medium to high risk, we are not going to use a diet which is formulated 100% on the jowar or 100% on rice cake. We're going to dilute these raw materials, so the contamination is going to decrease. So you might as well, at the end, if you're sourcing the other materials properly, you might end up with a low contamination profile. So that's why I picked up the minimum inclusion rate of half a kilo. And I'm talking about Mycofix Plus because we're talking about layers. So we are talking about the best in-class product that we have. I assumed a period of 80 weeks and a feed intake of 62 kg for the whole period with a 325,000 eggs per hen per house. And an egg price, I try to work out a little bit of a national average, because I know that the egg prices are varying from north to south, but I try to come up with this mid figure, and don't be too much into the number. This is just to give you an example. And I want to show you a few break-evens. So in order to break even for using half a kilo of Mycofix Plus, and again, the price, I kept a high price because I was keeping in mind about all the variations that are in the different distribution channels, and I picked up really a very high margin. But even with a high margin, we come up with a break-even of only 2.48 eggs needed more per cycle, per hen per cycle, or a mortality improvement of 0.8%. And those are results that can be easily achieved with the proper mycotoxin risk management, and can achieve with our products. And I will show you later a few trials that hopefully will explain you why. Doing the same exercise with breeders. Again, I'm talking about Mycofix Plus, the best in line product. Why are we talking about Mycofix Plus? Because layers and breeders are expensive birds, but we also have other products which are more cost effective in our portfolio. Again, I kept the Mycofix dosage at half a kilo per ton of feed, and I calculate this through a period of 65 weeks with a feed intake of 55 kilograms per bird. Also, this is the day-old chicks hatch per hen, so 150, and DOC price, which is 24 Indian rupiah based, and I base this calculation on national average. And to break even from using this product, all we need to have is 0.44% day-old chicks more at the end of the cycle, or a hen mortality improvement of 0.3% and a hatchability improvement of 0.29%. So really, really low numbers just to break even with this product. And again, this is a call out there not to use cheap binders, not to use on cheap products, because those products, they do not bring any benefits. I mentioned before the importance of stressing of using scientifically validated feed additives, and this is because we know, and you know, that they work and they're going to help you to achieve results. And we can guarantee that you can achieve those results by using our solutions. Let me show you how do this product work. Okay, so I want to show you very briefly a couple of trials, Mycofix Plus in laying hens. And again, this is just for 14 days, so very short trials. And what we have measured is the laying rate, average egg weight, and FCR. And we used a higher contamination scenario, but we also used a higher inclusion rate of Mycofix. If you have a look at the laying rate and the average egg weight, we have the group that only receive a mixture of mycotoxins, and the group that received the same mixtures of mycotoxins but Mycofix Plus. We had an increase in the laying rate of only 4.3%. And if you remember from my slide before, it was only around 2.48% needed. So we are observing consistently throughout all our trials a 4% improvement in the laying rate. So again, by using this product, because they really do the job that they're made for, they really deactivate the mycotoxins, you can expect to see those benefits. And also the average egg weight was also increased by 1 gram per egg. And the FCR was reduced by 10 points in the group that received the product. So this is a huge improvement if you think about, and this is money that are actually saved. One breeder trial that I want to show you, this is a combination of Mycofix Plus and BEP. This is a trial that has been done in my country, in Italy, on breeders. And again, we are talking about an inclusion rate of 1 kilogram per ton of Mycofix Plus, and also 1 kilogram per ton of BIOMIN BEP, and only from 18 to 46 weeks of ages. If we have a look at the mycotoxin contamination, you can see that this is really representative of field. So some of these numbers, you have also seen them in the tables that I showed you before. So we go from low to like few spikes, and all the main mycotoxins are present in there. That's the most important part. So low levels to in some cases spikes, and in the case of fumonisin, we had a little bit of a higher levels here. And if you have a look at the performance in breeders, you can see that by using the product and the combination of products, you can actually have a 1.3% improvement in fertility using 1 kg of Mycofix in a low contamination scenario, which you could have used maybe less. 0.5% in hatchability improvement and 2% improvement in born chicken per eggs, which is like a lot higher than what I showed you before in the break-even calculation. So again, just to stress the importance of using a product that we know it works, that we can guarantee you it works. I reached the end of my talk, so a little bit of a take-home message. So we have plenty of evidence, even coming from peer-reviewed paper. We have more and more peer-reviewed papers which are publishing effects of mycotoxins on poultry production at different stage at field levels. Importantly, and I've seen this from the materials that you submitted, so the long-living birds, so most of the material that you submitted are from long-living birds, and long-living birds are an investment and need to be protected. So the immunity, the growth, the nutrition, and the productivity. Keep in mind that alternative raw materials are indeed a cost-effective solution when the main grain price or the protein prices increase, but the mycotoxin load in some of them might be significant, and we need to keep that in mind to keep our productivity safe. A good deal of samples that we analyzed presented high mycotoxin value also that could harm productivity, so even in this small pool of samples. And as I showed you in the last part of the presentation, investing in the right mycotoxin counteraction tool can indeed protect the productivity. It can boost the revenue. So using something that works makes a difference, and the chip binder will not help to go through. I reached the end of this talk. Thank you so much for listening to me, and do visit our mycotoxins.info website if you want to learn more about the effects of mycotoxins in animal species. And again, thank you for your valuable time. I hope you enjoyed the presentation, and I give it back to you, Arun.
Yeah, thank you very much, Michele. You covered a lot of topics about mycotoxin survey and the mycotoxin risks involved with all these commodities. So we would quickly move on to the Q&A section. We have got a number of questions from the attendees. The first question, I would like to address it to Dr. Neil. Dr. Neil, it's a question from Dr. Pooja. So I will read out the question for you. Is there any chance that soya could be completely replaced with an alternate protein source by reformulating with specific amino acids?
Unfortunately, you can't really replace. Well, sorry. Yes, in many cases, if you have good quality canola meal, you can not use soybean meal at all. So there are situations where it is possible to completely use soybean meal, completely replace soybean meal, and use synthetic amino acids. It really depends on what raw materials you have available and the price in your country. Probably the better strategy is reduce the amount of soybean meal. The most commercially and cost-effective strategy is reduce the amount of soybean meal by maybe 20 to 30%, and then use alternative sources and balance those with amino acids, and that will give you still the nutrient value of the soybean meal because it is a very balanced source, and you don't run the risk of having this necrotic enteritis potential problem, and then take some advantages of the cost saving by using those alternative raw materials. So yes, it is possible to make a diet without soybean meal. It's done in many countries when soybean meal prices are really high, and we do it in Australia. But canola meal, we make Australia canola meal, and it is canola, not rapeseed, and it's a very good source. So it is possible, but probably in India at the moment with the raw material prices and restrictions, you might have to just reduce soybean meal. Might be the best option. Hopefully that addresses his question.
Dr. Neil, I think that was a great question and a great response to that, Dr. Neil. The next question I would like to address to Michele. Michele, after this steep rise in soy meal prices in India, the Indian government recently announced that they allowed import of GM soy meal to India. So how do you see this in the mycotoxin risk perspective? Genetically modified.
Well,
Soy.
I mean, as for the mycotoxin perspective, there isn't really a big change for the fungus if the soybean meal is genetically modified or not. And fungus, they have been around us for a million of years. They adapt. They are the most adaptable organism. And what we're seeing from trends worldwide is the mycotoxin contamination is increasing steeply. So this is largely to the global warming, to what's happening now on the planet, to global trading, and to this more severe and extreme weather effects. So I would think that mycotoxin contamination will keep rising, despite of all these commodities. And I think what makes sense for producers is to just always monitor. Whether using a new commodity, just analyze the mycotoxins, and as I mentioned before, try to prevent rather than act, and try to see the problem before it reaches the animal. So use a consistent protection and test.
Okay, thank you, Michele. Dr. Neil, I would like to bring you the same question, to get your opinion as a nutritionist or a formulator. Now, how do you see this, like GM soy? This is the first time in India. That's what, if I'm not wrong, unprecedented in India, the import
Okay.
of GM.
Yeah. From a technical point of view, and I've spent a lot of time over my career assessing and looking at the mechanisms that how it was genetically modified, there is no difference in the nutrient composition of the soybean meal because of the genetically modification. What happens with the genetic modification is that genes are inserted into the soy to make them insect and pesticide resistant, but the product is unchanged. The only thing is that you get better yield with the GMO, and so you get more soybean meal available, or more soy, and therefore more soybean meal. So nutrition-wise, there is no concern. The only concern with the GM going into India is a consumer perception and a market issue as to whether the people that are buying the broiler meat or the eggs will accept the GM. Again, I'm quite confident. I regularly would consume products that have had GM. I have no issue with the GM component because it's not affecting me. But yeah, markets, there are specific market barriers to using GM soy, and that would, the only restriction in India would be whether there's market perception rather than technical. Yep.
Thanks a lot, Dr. Neil. I would just like to pose one last question, and I would like to address to Michele. Michele, this is a question to you. Bit a long question, so please. If groundnut cake is being used as a protein source, then at what percentage toxin binder is added in the feed with the increase in inclusion percentage of groundnut cake?
I mean, what concentration the toxin binder should be used in the field? I mean, it depends really on the contamination that is present on the groundnut cake. And the only way to know that is to test the groundnut cake for mycotoxins and have a look about what's in there. The dosage of Mycofix is strictly related to the contamination that is in there. So we need a certain amount of product to tackle a certain amount of mycotoxins. So if we have a very contaminated groundnut cake with like lots of aflatoxin, fumarisin, ochratoxin, everything, and we are using maybe a big amount of it, and we are using only half a kilo of Mycofix, that is not the way. We need to first assess the risk. We need to see how much this mycotoxin load contributes to the overall formulation. So when we reach the finished feed, and so we need to know how much we are diluting this groundnut cake. And then after that, we apply the product based on the risk assessment. And as I mentioned before, the dosage is very flexible. It can go from as little as half a kilo per ton of feed to a maximum of 2.5 kilograms per ton of feed. There is no need to put more than that because we have done tests, and that's already plenty enough to take out most of the mycotoxins, including aflatoxin.
Thank you, Michele. So now we are at the end of our webinar sessions. So I would just like to thank you all for participating in this webinar. Of course, we have taken 10 more minutes more than the scheduled time. We really apologize you for that. But I hope that you all found this webinar very interesting and informative, especially in this present situation of soybean prices and the impending import of GM soy. So I thank you all once again. If you know our BIOMIN representatives personally in your region, you please reach out to them if you have any queries. Or alternatively, you can also contact us in our social media pages, BIOMIN India Facebook as well as BIOMIN India Twitter. And of course, our speakers have also shown their email addresses in their presentation slides, so you can very well contact them too. I once again thank you everyone, and thank you speakers Dr. Neil and Mr. Michele, and also I thank the Asia Pacific marketing team for their coordination. Thank you very much everyone. Stay safe and take care.